This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 171, recorded on February 17th, 2012. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today, right here in studio, Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
I'm doing great.
You were chipper yesterday.
I was chipper yesterday. Today I'm doing great.
Excellent. And how's the weather over there on your side of the desk?
It's as sunny as it is on yours.
It is nice, isn't it?
It's a beautiful day outside.
Beautiful?
It's a beauty of a day.
Let me check the temperature here on my
I would guess it's up to the 55 degrees.
50, you think?
I think.
Look, it says local weather, Silver Spring, Maryland. You know, these phones
It's not so local.
No, I swear it was last week. It's all messed up. It's not updating.
It's okay. It was.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How's it going, Alan?
Going well. It's 51 degrees Fahrenheit here.
Whoa.
I knew it.
And nice job on the intro, Vincent. I now know that that's easy to flub.
Oh, it's okay. I've been doing it a lot. 169 times I have done it. I think you've done it twice.
Yes.
So you guys did a good job. Nice show last week. Very interesting.
It was a fun time.
Compelling.
We all had fun.
Would you like to do it without me all the time?
No.
No, of course not.
The last time our next guest was on, well, I should introduce him, then I'll tell the story. Joining us today from Baltimore, Maryland, at the University of Maryland School of Medicine in the Department of Microbiology and Immunology, this is a namesake of our department, Dickson.
Yes.
Matt Frieman.
Hi, guys. How are you?
We're pretty well here, as you can tell. We're joking around. We're
It
happy.
is up 51 degrees and a little cloudy here.
Wow, 51 down there.
So it must be 51 here, too. So.
Yeah, my iPhone is
Right,
not
statistically.
refreshing.
But there is a heat bubble that forms over big cities, so perhaps it's a little bit warmer here.
Right.
Yeah, I can't get the temperature here.
Are you saying Baltimore's not a big city?
Well, not compared to New York, it isn't.
New York, of course.
Matt is a Baltimorean, right?
I am indeed a Balmer. Balmer bred.
That's a Balmer.
I grew up here in the suburbs and went away for school, came back for grad school at Hopkins, and back down to UNC Chapel Hill for my postdoc, and then back up here for my faculty position at University of Maryland. And
Cool.
Been here about 2.5 years, and it's going really well.
Yeah, you've done good for an Owings Mills boy.
There you go. Exactly.
He knows TWiV. You can tell. He gave me all the info.
That's right.
That's right.
It's pretty funny. So you've been in your lab there for 2.5 years.
Yeah,
That's
about
cool.
2.5 years now.
And you're funded, too. That's good.
We are. So I don't know what what kind of karmic things I did in the past, but we have been blessed with. I had a K-22 when I came in, which was a postdoc grant that I applied for. So that funded me for a little while with my startup money, and then we just were blessed with an R01 last summer. So
Congratulations.
We're doing good. I feel it gives a little bit of weight off the shoulders and lets us kind of really enjoy ourselves and get the lab going.
People really can win.
Yes, I wish I knew, because the next grant I put in was completely destroyed. So
Yeah, of course.
So the one you got, what was that on? Was that a SARS grant?
So it was a SARS grant, yeah. So we work on SARS, and we work on SARS basically as a model of acute lung disease. Right. And so we can use SARS to basically take apart lung function and to figure out how the lung responds to the virus, how the virus induces different pathways in different parts of the immune response, and then really try to figure out what's going on, and then potentially treatments. So
you're trying to ferret out the details of it.
Oof.
Exactly.
Do you use ferrets?
We do not use ferrets.
Do you use civets?
I was in Ralph Barrick's lab at UNC for my postdoc, and we actually had talk about using ferrets there, and we started looking into caging, and we actually had a ferret wrangler that taught one of our technicians how to deal with ferrets, but we never got around to actually doing it.
No civets either?
No civets, no.
What do you use, mice? You use mice, right?
Yeah, we use mice. So mice are our model. And for SARS, and we actually have a mouse-adapted SARS, so if you take the human strain of SARS and put it into a mouse, it doesn't do anything. It replicates. The mouse has mild disease, mild inflammation, but no real outward symptoms of disease, and the virus is cleared. But if what happened, I don't know how long ago now, but Kanta Sabaro at NIH, in collaboration with my old boss, Ralph Barrick, they made a mouse-adapted virus, which was passaged, as we've talked about on TWiV for the H5N1, same exact way. I guess it's not been redacted, so I can explain how it works. For SARS, which coincidentally is, I'm sure, how you do it for flu as well, you intranasally infect a mouse, and after 2 days, they took the lungs out, squished them up, spun it down, and then put that supernatant, which contained now virus that had been passaged through the mouse once, back into another naive mouse. And they did that over and over and over again until you got disease and death. So the first virus you work on took 15 passages before it killed a mouse. So this is in BALB/c mice, so little white mice.
Right.
And then the virus was sequenced, and then those mutations were put back into the infectious clone for SARS, which what my postdoc lab had specialized in. And we did a bunch of analysis to show which mutations were important for disease, which ones weren't, and then really use that as our model. So this mouse-adapted model causes really amazing disease in the lungs, which several parts of it really look like humans as well. So
what kind of containment do you do that under?
So this is all BL3, not BL3 plus.
Mm-hmm.
So I know you guys have gone over that before in lab on TWiV. I mean, but so in BL3 we have to wear a respirator, so a PAPR, and which basically is a respirator that goes around your waist. It's battery operated, and it blows HEPA filtered air over a hood, through a hood that you wear over your head. And we wear full TWiV suits, so kind of like FedEx envelope with arms and legs. And double gloves and booties, and then that's done in a room that's under negative pressure. So all of the air is sucked into the room from the hallway, and then it goes out the ceiling of the room to the roof of the building, it's then HEPA filtered and then put out into the environment.
It goes to Baltimore.
It goes to Baltimore, exactly.
So this is for both your mouse-adapted and the human strains. You have to do this BSL-3, right?
Exactly. So even though it's mouse-adapted, it still can infect human cells, so you would imagine that it probably could infect people as well.
You don't do transmission experiments, right?
We don't. So mice don't sneeze.
Right.
So you can't actually get passage from, or I've never seen a report at least, of a SARS-infected mouse transmission to a naive mouse.
Yeah. Of course, the virus transmits among humans to begin with, so even if you got it to transmit in mice, no one would block that, right?
Right. Redaction would not hopefully be in our future if we did that. So exactly. So we're trying to So it's going really well. We have a couple good grad students and new people coming in, and looking for postdocs now. So we're good. I feel very, very lucky in getting a grant and being in the position we are. I know a lot of people are not that lucky as scientists right now, and study section and getting grants through NIH is very difficult.
I was just on study section last week. It's pretty tough. So if anyone wants to do a postdoc on SARS, they should contact you, right?
Please email me right now. We're looking at postdocs, and we have a couple positions. So if anybody out there wants it, give me a call.
Yeah, I was visiting you in December of last year. It's a good group, good department as well. A lot of resources. They have the Genome Center there. Robert Gallo is across the street.
And it's a fun town.
And you have Bertha's mussels.
Yes.
We do. We do have Bertha's mussels. We have good crabs.
Yes,
and,
you do. And bad crabs, too. I know you can get both kinds in Baltimore. I don't know that firsthand, of course. But
I wasn't going to ask.
And I thought of a source for the journal publication for the other articles that you were referring to in Acta Redacta. I thought that would be a good new journal that we could start.
I like that. It's good. I think we need like a WikiLeaks for science, right?
Nice.
A Wikidacta.
All right. Well, we look forward to discussing future work on TWiV.
Absolutely.
With you. But today we're going to do some papers. There's a couple of papers that you recommended, actually. So if no one likes them, you can blame Matt.
Because we never would have picked these.
Of course not.
Of course not.
Before we do that, I just want to mention that today the WHO made an announcement of the outcome of its meeting on the H5N1 research. They had collected a number of influenza people over in Geneva. They had a 2-day meeting. And today they said this is through BBC News that I'm getting a summary. They said it doesn't make sense to publish the papers in a redacted form. It's not of very use to scientists to get that. They would like, in principle, to publish them in full, but not yet. They want to wait and do some risk assessment.
And broaden the discussion, I think.
Yeah. So we'll see what happens.
Because this was just a couple of dozen people at this meeting.
Yeah.
So, Matt, can I ask you, from the perspective of someone who's vaguely familiar with viruses, but I actually was in China the year after the SARS outbreak occurred.
Oh, okay.
Yeah, to advise their public health officials as to how to handle middle management-based outbreak data. Okay, because they were very poor at reporting to begin with, as you will recall.
Right.
And they lost about $60 billion worth of tourist industry dollars for just scaring the hell out of the rest of the world after they finally did decide to come out and announce what was going on. So, of all the people who were infected with the SARS virus during that horrible outbreak, how many actually became infected? And then of those, how many went on to die from that infection? What is the mortality rate from SARS?
So the official WHO numbers are around almost 8,000 infected and around 800 deaths.
So that's a 10%.
Now, how are they
Mortality
calculating
rate.
how many infected?
Right. So that was actually interesting.
No, that's the case fatality rate.
That's the case fatality rate.
No, I asked you
Right.
about the mortality rate. Just how many people got infected but didn't get sick versus how many people got infected, got sick, and died.
So there's only one study that I know of where they so there was a study that went back into Hong Kong blood banks
Right.
and looked to see how many people were seropositive and when. And I'm trying to pull that paper up now. Of course, I'm not going to find it. But
I just thought I thought of it because of this controversy of H5N1 because they're
yeah, I want to say that there was up through several. One of the interesting things actually that came out of it was that up till several years before SARS was even known about, there were seropositive people in the Hong Kong that had blood bank had positive samples for.
Right.
So I think it's there definitely were. There's going to be many more people that were seropositive and probably seroconverted than the 8,000 that were officially infected. And the 8,000 number is even itself small just for the epidemic because
Right.
the definition of being a SARS patient changed during the epidemic. So there's reports of the Chinese government basically moving the goalposts to be what's called a SARS infected person.
Right.
And you basically have to be, I believe, PCR and ELISA positive for SARS. And and there's going to be a lot of villages and a lot of places in the country where that's just not, you're not going to be able to see those patients.
Sure.
So it's clearly 8,800 is the reported number, but I would imagine that there's probably many more early on. And even in the 8,000 number is only the acute deaths.
exactly.
So one of the things that we're finding that's been reported and is seen in patients that are following up, that are followed up now that survived the outbreak is that they still have continued lower lung function than normal before they were infected. So I actually talked to there's a guy in the University of Toronto named John Patkai who I just talked to a couple weeks ago, and he's a respiratory therapist who is working with a cohort of 50 individuals that got infected with SARS in Toronto, and they were severely infected and they recovered. And no, he said no, not one of those individuals is back to their full lung function at the moment.
Interesting.
That was 2003 till now. So there are definitely a big cohort of people that have probably developed lung injury, acute lung injury, or ARDS, and died of pulmonary dysfunction since the outbreak that are not reported in those 800 at all. So the virus itself is very interesting. What it does to people is, I think, really worthwhile in looking at because it probably reflects a lot of what's going on in high-path flu as well. So whether you're infected with potentially a ferret passaged H5N1, what do those lungs look like? What do the people have, and how would they survive after the infection? So it's a good model.
Thank you.
So why can we publish SARS data
Exactly.
with impunity?
Exactly.
and we can't H5N1?
Exactly.
I don't know.
Yeah, it's a rhetorical question.
Because in the beginning of the outbreak, they thought the mortality rate was quite high.
Of the SARS outbreak.
Yeah, SARS. But I mean, even one of the people who discovered it, and like I say, he was in Vietnam. One of the physicians actually died from it.
Yeah.
Right. Yeah, Carlo Urbani. So the wild type strain of the human strain of SARS that most labs use in the U.S. at least is called the Urbani strain.
Right.
And it's actually the virus from him.
And then it spread to Toronto, for instance.
Spread to Toronto. Yeah, there was a single. It was actually a doctor who was visiting family in Hong Kong or in relatives in Hong Kong, and then he went back to his practice in Toronto in the hospital and infected patients and nurses.
Exactly.
So it spread.
Yep.
So the mortality rate of 10% is probably an overestimate.
Yep.
And probably it's the same for H5. It's an overestimate
Yeah, well,
as well. But they're both respiratory pathogens. They cause disease. So you would think you would be more consistent so I don't want to diss or I don't want to cause trouble for the SARS community.
Right.
I think it makes sense to allow you to publish everything, right? And
Yeah, as long as you work with it in a confined environment like you described, the
BL3.
BL3.
They don't have plus. It's just BSL3.
Right.
So H5, you have the additional containment because, well, for whatever reason.
Sure.
I mean, one of the big, the big differences, I think, the important difference between current H5 viruses in the nature and SARS is that SARS is already a human-to-human transmittable virus. So,
right.
No, the mutations we're putting in it, we're actually looking at things that attenuate the virus to figure out what the viral proteins do, not make it worse. And in the ferret studies for H5N1, you're taking a virus that does not pass animal to animal, at least mammal to mammal, and using a mammalian model to look at that.
Sure.
Whether that is important for human, as you've talked about, it's unknown. But I don't know if you want to talk about the H5 stuff now or not. But I think that the question about the model is interesting, and a lot of reporters have brought it up. Is that you can't have it both ways. Either it's a really good model, so give me money to study H5N1 in ferrets because ferrets are a good model for humans. So what I learn in ferrets can be used in humans. But then at the end of the passage experiment, we said, well, let us do it because ferrets aren't really a great model for humans. So
there are different things you can model. You can model epidemiology, which you can't model that in animals. Animals are not going to display the same characteristics as humans in that respect, or you can model mechanism.
Right.
And clearly animal models for mechanism, that's a long established field. And so it's a distinction between the different types of things that you could try to model.
Right.
I don't think there's a hypocrisy there at all.
Well, I think the distinction that was lost on the reporters and the science
it is lost on the general public, unfortunately.
Absolutely. Even Laurie Garrett.
Well, and part of the reason that it's lost on the general public is because of all this hype that surrounds, we found a new cancer treatment in mice,
right?
And those sorts of things. And scientists are partly to blame for that misunderstanding in the public mind because for years we've been simplifying this and saying, we model diseases in animals. We need to be a little clearer about what it is we're modeling in animals.
At WHO today, during the press conference, they said a large part of the H5N1 problem is a PR problem.
Yeah.
Yes.
And they're absolutely right. And Helen Branswell, a Canadian reporter, was surprised at that. But that's it, because we're not communicating well.
Yes.
At every level, from Foucher's initial comments to the value of animal models. Yeah.
Right.
Yeah, I think it's. We actually had Carl Zimmer, the science writer, here 2 weeks ago to give a seminar, and it was really interesting to talk to him about how to communicate science to the public. And there's things you have to leave out because it doesn't make sense to someone. The vocabulary is different, but you have to try to explain the important points and give those nuggets of information that are important. And if a scientist, if I was talking to someone in my family about SARS or this H5N1 virus, if I had to give them a 2-sentence explanation of what it was, it's really hard to do that. And you have to, science writers write for that first paragraph of the article, and you try to keep them in for the rest of it. So you have to, they write it as a hyped kind of story, and it's hard to get to this nugget of science in that story. And I think that's really what separates the good writers from the bad writers. And once one of those hype stories starts getting around, it just it gets in the papers and then runs with it. So
ends up in the echo chamber. Yep.
Yeah, no one wants to read a boring article about why this really isn't a bad virus when the scientists that did it said this is a bad virus.
Right.
I think that's a big problem.
Yeah,
yes.
but you can see what happened to SARS after the tumult settled down and the dust cleared and the true facts emerged. It's more reasonable to look at SARS as just another nasty virus that could sometimes come back, but if it does, we're prepared for it, and we are not afraid of it like we used to be because we didn't understand it. And understanding comes from knowledge, and knowledge comes from doing the work and publishing. And so
And fear comes from lack of knowledge.
fear
right?
comes from lack of knowledge. There's no question about it.
But it's still a good virus to fund at IH. So
Of course it is.
there's still a lot we don't understand.
No, but you can separate out safety issues from whether you should even know about this or not. Those are two separate issues.
Sure.
Exactly. Yeah. No, they are two separate things. And I think that I think the pause in the H5N1 research with all of the hype that happened after the first couple stories were out is probably warranted. And once everyone understands what it's really in the papers, and if anyone ever gets to see the papers, it will be probably less exciting and crazy as it sounds.
Right.
Yeah.
Totally agreed. Maybe not even reproducible in another place.
Well, that's science. You have to be able to show that it's the same. So in two different labs, and if you don't know what the mutations are,
Exactly.
you can't do
do
the
that.
science. You can't do it. Otherwise, it's just one guy screaming from a rooftop, and that's it. So
Yeah.
we need to be able to do that kind of work.
Wouldn't it be amazing if after all this, it's published in 3 months and nobody can reproduce it?
Exactly.
I mean, this is just
Yeah.
crazy. I mean, I guess it's an in-principle discussion, which is fine, but that would be
Right.
just amazing.
Yeah.
But I mean, the people that want to stop the research and block it are doing it, theoretical. In principle, this is the terrible thing that's going to kill the world anyway. So, I mean, you have to balance the fear and the safety issues with actually getting knowledge out of the work. And I think the 1918 flu virus is a great example because there's been an immense amount of understanding about flu biology and epidemic flus from that research. And initially, that was a really scary virus. And once people started looking at it and actually doing work on it, it became a lot less scary.
Yeah.
And so, I think that there's still incredibly good scientists and a lot of information that for human health that can come out of the H5 work. And I do think that once all the craziness and the scare mongering and the fear mongering settles down, it will be a much more intelligent conversation.
Here, here.
Yes.
We'll see. We'll be here.
That's right.
We'll talk about
That's
it. In fact, we have some letters about it later, so we'll revisit this.
Yeah.
We have a couple of papers, interesting papers. The first one is from Tim and Yin in Virology, Kinetics of Virus Production from Single Cells. This is from the Wisconsin Institute for Discovery. I like that name.
Yeah.
Great name at the University of Wisconsin-Madison.
And I love this paper.
I think it's cool.
This is so awesome. They used, I don't think there are any 21st century techniques in here. and yet they did something really, really cool.
Yeah, you're right. So they basically want to know, when you study viral growth in cells, you do a growth curve. You infect a plate full of cells, several million cells, and you see what happens in the population. You can monitor the production of virus over time, how much is made and when. But what they did here is they want to know what happens in an individual cell.
Right. So normally we're looking at population behavior, but what's going on individually? Are all the cells really behaving the way the whole population does?
So they do a very. I was amazed because I thought there was going to be some high tech stuff in here. They
When
just do
when
limits.
I saw the abstract, I thought, wait, you what?
Limit dilution. They make dilutions of cells. So they actually infect cells. They use this.
Why
is
the hell did you think of this?
The virus is vesicular stomatitis virus. It is BHK cells, I think. I remember.
Baby hamster
Yes,
kidney
I think so.
cells.
And they infect them. Then they make dilutions and put a dilution in each well of a 96 well plate, such that most of the time you get no cells, and now and then you get one.
Right. So it's serial dilution. That a 19th century technique, right?
Yeah.
At least.
And then they look under the microscope to see which wells have one cell, and they mark those. And then they study the production of virus. They take time points out and they do a plaque assay to measure virus produced.
Yeah.
And this gives you some hint on how diverse this population of cells is, because in the end they find that the amount of virus released per cell varies quite widely. And when it's released also varies quite widely, which is not something you would know by doing single-cell kinetics.
Yeah, I think the timing of it was the interesting thing for me. I was really surprised at how variable in the time there was.
Yeah.
Yeah.
Can't you also say something about the variability of the viral particle itself, too? You can't assume that they're all identical, can you?
Well, they go into some of the possibilities to explain why this might be, why you might get such different growth behavior in each individual cell.
Right.
And one is that the individual particles are differing in their fundamental ability to replicate.
Right.
Right.
The other interesting thing is that no matter how much you infect with, the yield is pretty much the same per cell. Mm-hmm. So the multiplicity of infection, they tried 5 and 15, I think. Yeah.
No, they do more. They do 5, 15, 20, 30, 40, and 80 MOI.
And they're all pretty much the same yield per cell, right?
Yeah. I mean, it varies maybe threefolds. I mean, that's
so
max. So
that brings back the colored virus story of from Lynn Enquist's lab, which we did last year. If you remember, no matter how many herpes viruses you put in a cell, only 5 to 7 seem to actually replicate. So this result would imply that no matter how many you put in, you can only get a limited number of replication sites in a cell.
Right. The rate limiting step is not the number of particles that come in.
Yeah.
Or is not related to the number of particles that come in.
And that's basically it.
Right? Yeah, and another possibility to explain these differences that they saw, they point out you could be infecting cells at different points in the cell cycle.
Right.
Right.
Yeah, this is a question that Dickson and Steve Goff were arguing just before we came on here, right, Dickson?
Well, we weren't arguing. We were just...
You were wrestling on the floor.
Nothing wrong with arguing.
No, we were positing what the next set of experiments should be and how to conduct them. And
what did you decide?
Well, Steve
I would
was
want
a
to
big
do this
help.
in synchronized cells.
Exactly.
Right.
Yeah.
And you can then infect them at various stages of the cell cycle and see A, susceptibility factors, and B, viral titer production, as long as you can be sure that the virus that you're giving is homogeneous. Which is that a big assumption? I don't know, guys. I'm not a virologist.
Genetically homogeneous?
Yeah, or functional. Just whether all the virus particles are functional.
They're not.
No.
So what proportion aren't? Is it
What
a
is it
variable thing with virus species, or is it?
Yeah.
Yes.
Okay.
Yes, it varies.
So,
tremendously.
okay. What about the next question then? What about for this virus, VSV?
Yeah. So the particle to PFU ratio is quite variable for all viruses.
And so, but how would you test that unless they did it on a synchronized cell?
So what does that mean?
How do you know that they're that way? It could have been for this reason, not the other reason.
I don't know what you're talking
Since
about.
nobody has done this experiment with synchronized cells that make them all the same, and this result shows differences in the production per cell, how do you know that your virus particles are different rather than it was just a difference in cell cycle?
Well, because the way you do a particle to PFU, you count the number of particles in a prep by EM, for example.
Go on.
And then you do a plaque assay and you count the number of plaques.
Right.
And you assume that all of them got into the cells?
No,
No.
you're looking at population averages in that case.
Yeah,
Exactly.
You don't make that assumption.
Exactly.
And so you're looking at the population average to get the number of plaque forming units, and then you count the particles under EM to get the number of physical particles. And as long as it doesn't really matter that the cells are whether the cells are synchronized or not.
So fewer plaques versus more particles.
Yeah.
Right.
So for some viruses, Semliki forest, it's one each. Pretty much each particle is infectious. For papillomavirus, it's 10,000.
Really?
1 in 10,000 is infectious. Yeah.
Wow. What about polio, Vince?
Polio is between 30 and 1,000.
Can you engineer them to all be the same?
No.
No.
So
But you can-
why
you can
aren't
No,
they?
actually,
Can't you?
that was an issue that I struggled with in my thesis research on polio entry.
So the question then is, what's the difference between a viable particle and a non-viable particle?
Good question.
Darn good question.
See, every step.
Alan, did you find out?
No, actually, I developed some interesting leads on how the virus is getting in, but I can't tell you
This is interesting.
what is making something a defective particle and what's.
This is
We
very.
think that basically there could be mutant particles, there could be broken particles. They
Or
could packaging. They could fail at different steps of the cycle. It's quite complicated to get through this whole cycle.
Or maybe that's not a good cell for that virus.
Well, that's a good question, whether the particle to PFU ratio differs in different cells. It probably does. I'm sure there's a cell contribution.
And this is also done all in cell culture with a single type of cell. So
Right.
what happens in an animal or a host where
Sure. Different
you have
cell types.
different
Yeah.
cells, and depending on what
Exactly.
cells the virus can infect versus not infect, bind to but not actually produce a lot viable
Wait, Vince
infection.
is getting an idea, yes?
No,
Yeah.
why don't we infect with single particles, Dickson?
That's what I was going to ask you next.
Well, we actually have a paper with.
You do a single
Later.
Talk
cell
about that.
with a single particle and see what happens.
Yeah, that's I would think the single virus might be doable when you hear one of the next papers we talk
Now,
about.
I just finally dug up particle to PFU for VSV. Got a paper from 1976, and they found between 10 to 50 particles per PFU.
Not bad.
I think one of the other things that was interesting in this paper was that they did a latency time period graphs where they looked at how long it took for the virus to start being produced from each cell.
Yes.
Yes.
Yeah.
And that's variable as well.
Yeah. Yep.
Yes, but I think that they found that the longer it took for that first bump of virus to come out, the more virus you actually produced. So
That's
like
interesting.
if you look at the slope of the curve,
Yeah,
if
yeah.
you got more virus, the longer it took. So I don't know really what that says about if it's a cell cycle issue or it's the virus making a bunch more viral machinery and particles inside the cell before they really start coming out.
Right.
Maybe depending on the cell cycle, you can make a lot more viral products and subunits and viral proteins inside the cell before you actually produce it because there's something in the cell cycle that blocks that release. And then all of a sudden you have that big release. But I don't know. I think all those questions are interesting.
The cell cycle experiment. So Steve Goff was telling us an easy way to synchronize cells. He
Yeah,
said,
this
what
is
you do
good.
is you take a monolayer, and every so often you rinse it, because when the cells divide, just when they divide, they're detached from the monolayer. So you can wash off all of those cells, and then you'll have them all synchronized at that phase right after mitosis, right after cell division.
Clever.
Isn't that cool?
That's good.
I mean, there are other ways too, but he said that would
Yeah.
be that's a really nice way if you have a lot of cells to do that.
And you could automate that.
Yeah, you could.
You could do that.
Should we do this with polio, Dickson?
Gee, Vince, it sounds like it's a possibility.
Yeah, synchronize them up. So that's people have asked that question for years with RNA viruses. What's the effect of the cell cycle?
Yeah.
For DNA viruses, many of them require the cells to be in S phase, synthesis phase, where they're replicating DNA, because the DNA viruses use the host DNA replication apparatus.
Right.
So it has to be active.
Yep.
In fact, many viruses, DNA viruses, when they infect cells, they kick them into S phase just for that reason. Isn't that interesting, Dickson?
Definitely is interesting, Vince. I'm sitting here with my eyes wide open. I'm even thinking back to some early experiments that the Israelis did when they discovered the West Nile virus. They were using mosquitoes in this case, and the temperature varies in the outdoor environment, and the titer of the virus that accumulates inside the mosquito varies with the temperature. So I'm just wondering whether that could be applied to this kind of a situation. I know this is all at 37, but some of these animals have different temperatures than we do. Like if you used bird cells, for instance, you would have to use it at around 41 degrees, and a lot of the viruses that we use don't replicate at that temperature, right? That's
Is that right? 41 degree for a bird cell?
Yeah.
Wow.
That's their normal body temperature, so you'd have to match that in order to get the conditions right for their particular set of viruses.
That could be a major barrier to adapting a bird virus to mammals, too.
Bingo! And what are we talking about here?
Yeah.
Flu virus, right?
Anyway, we should, we shouldn't make this week in flu.
No, we shouldn't, but one flew where? Sorry.
Now, when Matt had uncovered this paper, I immediately remembered a classic paper done on polio, and then I looked it up, from 1955, from a group at Caltech. The first author is André Lvov. Then we have Renato Dulbecco, Marguerite Vogt, and Marguerite Lvov. Now,
I've heard of some of those people.
Dulbecco and Vogt had just developed the plaque assay for animal viruses. And so presumably Lvov came over to do some plaque assays in his laboratory at Caltech. Lvov, of course, went on to win the Nobel Prize in 1969 for discovering lysogeny of bacteriophage lambda. But here we are in 1955, the name of the paper, Kinetics of the Release of Poliomyelitis Virus from Single Cells.
Didn't Delbecco also win it?
Delbecco won it later
did.
as well.
Later.
Yeah, with Taman in Baltimore in 1970s for tumor viruses.
Did you see what issue of Virology this is in?
Number 1. It's the first issue. It's page 128. It must have been like among the first papers. And the funny thing is, so they're studying kinetics of polio infection in single cells. The way it's written, we have gone over some old papers, and the way they're written are so different from now. This sentence, although our study of isolated animal cells was started only a few months ago, the results obtained so far have been encouraging. We feel that these should be published now to stimulate further investigation. It's just something you would never see in a paper today. It's just funny. So what they do here, they use a different way to separate single cells. They basically use little tiny pipettes that they've drawn out to get single cells, and they put them in a kind of a hanging drop apparatus. They make a complicated apparatus with oil and hanging drops. But basically they can take individual cells that have been infected and put them in this apparatus and then measure the virus production. And in this paper, they do 4 cells. Right?
A total of
4 different cells.
right.
They have had infected with polio, and they have a table here, number of PFU released by cell number 1 and cell number 2. And the interesting thing is, what they say is everything is released within an hour. All the virus that's going to be made is pretty much released in an hour. And when it's released varies. So here in this table, it's first released at 6 hours post-infection, and then the other cell, it's released at 7 hours. In one cell, it's released at 5.5. And so the time of release goes from 5 to 7 hours, and the amount of virus is varied also. So it's very similar to what we've just talked about.
Were these cloned cells, or were these isolated from organs?
These were cell lines.
Even then they had cell lines.
Sorry, one of them was HeLa cells, and one of them was primary monkey kidney cells.
Right.
So HeLa's had just been, of course, developed not too many years before this.
Yeah,
So this
53, right? So that's
is
like the paper 53, and this is 55.
This
so
is
it's very soon.
cutting edge stuff.
And Delbecco's medium was probably being used also to grow them.
probably.
Yes.
So I thought it was cool that
Amazing.
way back when we sort of knew this, but not much in between.
Right.
I have a nice picture of the changes in single cells that go on when they become infected.
So, Vince, here's a question for you. If you're designing this experiment today, and if you could correlate viral replication cycle with capsid production, and if you could label the capsid protein with fluorescent green protein to begin with, could you actually watch the virus being made per cell in real time?
Sure.
Yeah.
So why hasn't somebody done that?
I don't know. What I would do, though, I would infect cells, then I would sort them with a flow sorter and put one cell in each well.
Right.
And then go back to the lab and watch them under a microscope.
Yeah. I mean, you could actually make movies of this.
'Cause, that way you get one cell per well. You don't have to do this.
Exactly.
Look for the wells with one cell.
Sure. So in that way, you could actually measure viral production rate in a colony of cells, even though that wasn't homogeneous in terms of its growth cycle.
Sure.
And in another way, you could put another fluorescent protein in there, which would indicate at which phase of the cell cycle the cell was at. So you could get like a pink color for the cell cycle and a green color for the viral particle production, and you could have this beautiful movie playing.
You could do that.
in
You could even have a dye for the RNA, probably.
Yeah, you could
Right.
do that.
Yeah, you could do all this. Want to write a grant, Dickson?
No, I want to make the movie. I want to see this movie playing on the big screen.
Now, if you remember the plaque assay movie that we talked about a long time ago on TWiV.
Right. That's what I was thinking of too.
One of those versions, they had GFP fused to a late vaccinia virus protein.
Right.
So what you saw in the movie of the plaque, you saw the cell refractoriness change as the plaque moved outwards.
Right.
And then later, the green came up
Ding, bingo.
because it was a late protein.
Bingo.
Isn't that cool?
Very cool.
Yeah, neat stuff.
Very, very cool stuff. That's right.
And then the last sentence of this paper. To ascertain more details of the mechanisms of virus release in this virus cell system, many more cells will have to be studied.
Like 8 instead of 4.
That's right. At least enough to do students' t-test on it.
And then finally, a preliminary report of these results were given at the conference on polio at the New York Academy of Sciences, January 1955. Look at that. All right.
Yeah, cool. I really like reading the old papers. I had never seen this before, and I mean the language in it is, I think, amazing. It's much easier to read, I think, than our current papers we have to write.
Well, things were a lot simpler.
True, although at the time it didn't seem simple.
No,
right?
Of course not.
No.
Yeah.
No, but the way they describe how you prepare the glassware and you prepare the cover slips.
Sure.
And,
They washed it with soap.
The optimal degree of washing.
That's right.
Actually,
And
they
spreading
said
of aqueous medium has to be learned by experience.
If they said if you use soap, Dickson, it didn't work right because the drops spread out too far if you have
Right.
soap.
You had to use distilled water and alcohol.
They have a whole procedure, and they have this anecdotal stuff. It seems like this works a little better.
Yeah.
All right. Now we're on the topic of singles, right?
We are.
Single cells. Now we're going to move on to sequencing the genome of single virus particles.
Wow.
There you go.
I'd missed this one, Matt. Do you keep an eye out for cool tech stuff?
Yeah, every once in a while. As you saw when you came here, we have a big genomics institute, the Institute of Genome Sciences. So I'm always trying to give them a little more kind of virology to be able to use all of their new technology. So when I see something interesting, I kind of throw it at them. And,
come on.
And
You
this
were
one
just
came up, and
cruising
I really think it's pretty amazing you can do.
This is it. So the idea here is when we sequence most of the viruses in the world, you can't culture, so you have to sequence. You do mass sequencing to figure out what's in the ocean or what's in dirt or whatever, what's in your gut. And what they're saying here is they would like to know individual particle sequences, because that way it probably is easier that you don't have to pull out the sequences from a big database, I would guess, or data coming from an experiment. So, Matt, why would you want single genome sequences?
Oh, hello?
Yeah.
Yep.
Sorry, I lost you there. So people have used kind of single cell genomics for bacteria and all the metagenomic analysis to understand non-cultivatable bacteria species and microbiome. And I think this really gives you – there's a whole nother biome out there that's the virome, and understanding how non-cultivatable viruses what's there and what you can see by cell culture and what you're missing is important too. So this gives you a way to really start looking at the non-cultivatable species that you're completely missed by what can grow on a cell culture dish and what can't.
Right.
But if you take, say, a sample, you can do mass sequencing, deep sequencing, and pull out the individual genomes, right?
Right.
But this is just easier because you don't have to resort to bioinformatic trickery.
Right. When you're doing I've done a study with Eric Donaldson, who you've had on here before, about all of the bat metagenomics, looking at novel viruses we can find in bat poop, and there it's a big bioinformatic push after you take a sample and it's all a big mixed species there of everything, of all the RNA that's there, say. And this, you can take a single particle and really sequence that, and you can get a much, potentially. I think it's more people do this, maybe it'll get easier, but a much cleaner understanding of what's really there and what the sequence is, and get you on your path farther.
I see. I see.
Eric's done really well putting these viruses together we find in bats, but this is another way of doing it.
And this would also enable you to look at the amount of variability in a particular virus.
Yeah. Okay.
Sure.
Which is one of the question marks that you have to deal with the bioinformatic approach.
Yeah.
Right. And one question that's been talked about, talked about a lot when I was in North Carolina was, and we've kind of hit on it, that, when a virus is produced from a cell, you get a big swarm of viruses. So especially RNA viruses, you get mutations in the genome that occur at every replication cycle. And understanding what that swarm of viruses looks like, and how many of them are actually infectable, or infectious, and how many can go to form a plaque and infect another cell or not, is interesting.
Yeah.
And it helps us understand more about how virus species work. And so if you can now start sequencing those individual virus particles, you can start appreciating all the variability that occurs during a single round of infection.
The other issue, I guess, is that with deep sequencing approaches, you don't get long runs. So that is long stretches of sequence. So it becomes more difficult to assemble big genomes when they're novel viruses and you don't know what goes with what, right?
Exactly. No, that's actually true. And the technique they use here, MDA, multiple displacement amplification, uses a really, really long reading polymerase. So you get much longer fragments than you would at typical, high throughput Illumina or 454 sequencing.
Yeah, this is actually a really cool technique. This was one that I hadn't really paid much attention to before.
So they actually use flow cytometry to isolate single virions. And they found that staining them with cyber green, which is a fluorescent dye that binds to nucleic acids, then allows them to put them in a flow cytometer, which will sort them on the basis of this fluorescence.
Right.
Yeah.
But they don't do the virus themselves, right? They stick the viruses, or the viruses bind to a bead, an agarose bead, when that's the thing that's sorted. So it's like a carrier molecule, essentially.
Okay, so I was wondering how they sorted something as small as a virus. So it's attached to a big bead, yeah.
Right.
So it's attached via, how is it attached to the bead?
They use PTFE, polytetrafluoroethylene, they use cooled agarose beads that then bind to slides that have the PTFE on them. So it's just an agarose bead, I think, that's
sticks to
they're sticking to, yeah.
No. And then they put these on this PTFE slide, which has little wells in it.
Right.
So you put one virus particle per well, and then I think they cover it with agarose.
Yeah, and there's this cool figure 3 I thought was kind of cool with showing this little tiny blip. Here's a single virus in a agarose bead.
Yeah, there's just one there. Wow.
I thought it was a little overkill, but I love this single peak of fluorescence when they scan as a z-axis across.
So now you have single virions in these little wells, and then they sequence the DNA in situ.
That's wonderful.
Right.
And they use this technique, which I have not heard of before, this, what is it called?
Multiple displacement amplification, yeah.
MDA.
Yeah, that's what I thought was cool.
So it's using a DNA polymerase from a phage, a bacterial phage called Phe29, which you don't need to do any denaturation and renaturation. There's no high and low temperature cycling as you would do with PCR, because the polymerase, as it makes the product, as it copies the DNA, it displaces the new strand.
Yeah, and this polymerase is apparently super high fidelity and super processive, so it just chugs along for kilobase after kilobase, and it's got error rates that were something like 10 to the minus 7th.
Hugely low, right? As opposed to 1 in 10,000 for...
Now why Phe29 needs a polymerase that does that, I'm not sure, but...
It's a good question. So they make a lot of DNA from these individual virions. They use random primers to prime the polymerase reaction, and then they can make micrograms of genomic DNA, and then they sequence that by 454 or Illumina type sequencings.
And this is a continuous polymerase operation. It's not PCR.
Right.
So you don't cycle the temperature, you just incubate it at a steady temperature, and this polymerase goes in, and wherever one of these random primers stuck, it starts extending, and it just keeps extending.
Have you used this, Matt, this technique?
No, I never have. I like the idea, though. I think I know. I want to say I've heard people using this for single cells from human tissue, where you can cut out a single cell, amplify the DNA from the single cell to be used for arrays or deep sequencing or what have you. So I think this is that technique as well. But now you're stealing it and putting it onto a virus, which is, I think, really cool.
Yeah.
So in this paper, they do a pilot where they mix phage lambda with a T4 phage, and then they show they can get the lambda sequence, basically. They get the whole genome, and the coverage is like 450x or something. What does that mean, Matt, when you have 450x coverage?
So you're basically sequencing the entire genome 437 times, coverage. So at every base pair, you have, on average 400 reads for that single base.
It's amazing. And they say they got everything but the last 5 bases, which is funny.
So they didn't touch all the bases.
Yeah, out of 35,000. What was the genome size? Something like that.
48,502 base pairs.
Amazing.
And they got between 0 coverage, so I guess it's the 5 base pairs they lose, and 2,000x coverage. So, sometimes though the first 5 bases of the genome of viral genomes that are pretty important for further studies, but you figure that out later. This just gives you a full coverage there.
Yeah, so basically with this technique, you have to go to the ends at the end and make sure you've got everything. You do some primer extension reaction or some sort.
Right, that's something, though, this is obviously a proof of concept experiment. We already knew what the sequence of lambda was, but this, to me, it seems like the type of thing where you could just take any sample and now you can sequence single virus particles out of it and get just the potentially very clean, useful genome sequences that you would never have seen before.
Yeah,
right.
Absolutely. I mean, I think it for the type of viruses, especially the ones you can't culture. We could take one of our bat fecal preps and isolate the virions from there and do the same prep there, which, things that we probably maybe couldn't see at that depth before. So, and with you guys, you've touched on all the high throughput sequencing and the deep sequencing before on TWiV, but you can then take, the way the sequencing works, it's called barcoding. So you can take a single one of these cells, one of these wells that has a single virus, and then you can do all the amplification in that well, and then combine 200 samples together to be run all together, but every single one of the samples has a different little tag on the end. So you could then separate them all out once you get all the data and get 200 genomes all at the same time, which I think is pretty amazing.
So the barcode is part of the primers that you use for amplification, right?
Right.
Yeah.
And actually, I wonder if you would kick those off from with 529 or not.
That's a good question. Yeah.
I don't know if that would work, actually. But still, you could get the non-cultural viruses that you would normally never be able to see, which is really, I think, amazing.
Someone should do this on sewage.
Yep.
And ocean water.
Yep.
I have another question that doesn't relate exactly to this, if you'll allow me to ask.
No.
Okay, fine. The heck with all of you. There's a lot of DNA in thumbprints and fingerprints, and forensics is always trying to pin down who made that fingerprint for various reasons, crime scene investigation, but also to give provenance to art, for instance. Saw a nice Nova show the other night about a potential Leonardo da Vinci painting that had his thumbprint on it, but they couldn't identify the thumbprint because it was smudged. But it occurred to me, if they had varnished over this, it might have preserved the DNA on his thumbprint. Could you use this technique, because it's so sensitive, to actually get partial sequence that might allow you to identify, or at least to keep straight all of the other known Leonardo da Vinci paintings to this one that you think might be a Leonardo?
I think this this technique, they say, at least in the introduction, it seems to imply that it's already being used for single cell analysis in other systems.
Right.
So this was just extending it to viruses.
Right.
MDA, you mean, right?
Yeah, MDA. Yeah, right.
And this work is out of the Craig Venter Institute, as you might guess, in San Diego. Sequence. This is pretty cool. Cool tech.
Yeah.
Thanks for bringing that up, Matt.
Sure. No, I think all of these things, once you hear about them, then you start thinking about all the other cool things you could do with it, and you never know where you can get to.
Exactly.
That's why you have to publish stuff, right, Matt?
Exactly.
Yes.
Was that a hint? I'm working on it, I promise.
No, not for you,
No,
Matt.
not for you. No.
For a general audience we're talking about here.
Publication is essential in science.
That's right.
We have one quick story that Matt sent us because it's a local thing. It takes place in Maryland, although it actually has more far-reaching implications. This was an article in the Washington Post. Deadly virus hits turtles and tadpoles in Montgomery County. Where's Montgomery County? Is that where you are, Matt?
No, it's south of here, kind of outside D.C.
So
Montgomery County is to Maryland what Westchester County is to New York.
Got it.
There you go.
That's a great analogy. So basically, these box turtles and tadpoles are being killed by a virus called ranavirus. And in fact, there is a global epidemic of ranavirus infection of these cold-blooded animals. A lot of frogs in England have been dying of this virus. We actually talked about it a long time ago on TWiV, I think. These are rather large, double-stranded DNA viruses. They're in the family Iridoviridae, which you may recognize because some iridoviruses have been implicated in colony collapse syndrome in honeybees.
In bees, right?
Bees, right? Big genomes, 150 kb long, and they're killing these little turtles and tadpoles, and we don't know where they're coming from.
Now, I think ranavirus as well, based on the name, these are most associated with amphibians, but
Right.
I guess the reptiles and fish in the same ponds have been considered reservoir hosts.
Yeah.
But in this case, a big part of the news is that the virus is killing the turtles too.
Yeah, that's right.
So they're not just carriers.
That's right.
And by the way, this research was done at my alma mater, Towson University.
Really?
Oh, I didn't know you went there.
Yes, I did.
Very good.
Nor did
Back
I.
when it was Towson State.
Exactly. That just changed recently.
Yeah.
No, I mean, I think that I think this is a good, it connects with all of our bat stories as well because it's another one of these kind of sentinel animals that you never would really think would have a problem, and by disrupting the environment, and maybe that's leading to their the exposure of them to novel viruses and novel pathogens that they normally wouldn't see.
Right.
And this is all of these were found across this thing called the Intercounty Connector, which is a new highway that was built in Maryland that goes east to west across places where it was hard to get across from these two counties outside of DC. And who knows how the interaction of this big giant highway going through a place that was ecologically kind of blocked from development before is really affecting everything. So, another one of those things we need to look out for.
We'd call
Another
that,
sad aspect of development in Maryland.
call it fragmentation of ecosystems. That's what the general term is. Yeah.
And this, in the bigger ranavirus story in frogs, it seems like environmental stressors have been a big part of that story.
Yep.
Because these are not
That's right.
these are not some brand new pathogen that frogs had never encountered in their history before.
Right.
Right.
This is something that's been there in the environment, and there are a number of studies apparently that have found there's a substantial exposure rate. You can just catch a bunch of frogs and analyze them, and you find these perfectly healthy frogs, and some of them are infected with ranaviruses.
Yep.
So it's normally maybe not such a bad infection, but then the frogs get stressed by water pollution or water temperature changes or pesticides or other
UVB.
or a highway being paved through or increase in UV light. And then all of a sudden it becomes a problem.
Yep.
So in this study, they had put radio transponders onto the turtles. They had glued them on.
Right.
They had, I think, done 50 or so, and they found 31 dead near the construction site, but only 3 had been hit by cars or other things. The rest were just dead.
Right.
And that's the virus there. They were surprised. This guy said it's very unusual. 27 dead turtles in a 2 to 3 year period.
Right. Apparently, these particular turtles, they don't have any predators in the area.
Mm-hmm.
So they just live their natural half-century long life, and they die of natural causes. But this, all of a sudden, you've got this relatively massive die-off.
Yeah.
Gentlemen,
I love the picture of the turtle in the article with the transponder
yeah.
on his back too. It's great.
I think Dickson should leave.
I have to leave. Unfortunately, I do have to leave, and I don't want to miss this conversation, but I have to leave. So please accept my apologies for bailing out early today.
Okay, Dickson. Goodbye.
All right. Goodbye,
Take
gentlemen.
care.
See you.
Good to talk to you.
Yeah, same here.
Don't let the door hit you as it closes. You want me to give this as your pick later? All right. Very good. Thank you, Dickson. There are quite a few articles on these ranaviruses, and they're trying to figure out where they came from and, as Alan said, why the infection is lethal now. They seem to be more prominent in areas where cattle graze and the water quality is poor, so who knows what that's about.
Pesticides seem to increase susceptibility to them. Whole bunch of different factors. It almost seems like just about anything can
Right.
Yeah. Translocation of infected amphibians through commercial trade, food,
Right.
fish bait, pet industry contributes to the spread. So,
Right, that's another thing. That apparently they spread easily within a body of water, but wouldn't necessarily travel between them unless somebody
Yeah.
catches frogs in one place and uses them as fishing bait in another, or what have you.
Also found an interesting summary at savethefrogs.com.
Yeah.
They talk about this, so we'll put a link to that in the show notes. They compare it to the chytrid fungus, which is killing frogs as well, right?
Right.
And other amphibians.
And I think I think the interesting part of the story for me was that it really shows that ecological virology and ecological microbiology is important, and probably don't get as much of a funding level as medical human diseases do. But I think it's important for everybody to really know what's going on here, and it's to look to see that whether it's a human pathogen or not, and how it can move to animals. And, if you start affecting one species, then you can really affect all the other ones. We're all connected.
Unfortunately, at a medical school, it's hard to work on this kind of virus for the same reason that not many people work on plant viruses at medical schools, because the focus tends to be on human disease. But
right.
It's important stuff.
Yeah. I mean, as a model virus, these things are important, and I know, there's no reason not to, and to push it harder.
All right, shall we do some email?
Sure.
Sure.
I'm going to time this in a specific way so that Alan gets to read this third one. So, Alan, can you do the first one?
Oh, okay. All right. Tom writes, his subject is a non-scientific hypothesis to explain H5N1 claims. Hi, TWiVarians. Old trick to find enemy activity in an area of scientific research. First, claim a useful result that just works but really doesn't, and then trace the enemy signals when they test it. Imagine it's possible to monitor ferrets or some other signal. First, you track ferrets going to the bad guys, and then the voluntary moratorium and higher BSL requirements cut down on the noise. Big downsides for scientists in this scheme: you can't promote a good idea, or the enemy might make it work, and you need a way to drop the idea, or you risk becoming a Duisburg. Thanks for the enthusiasm, conversation, knowledge, ideas, and great audio. My health span will end someday, but with your help, it won't be from a microbe.
Cool.
So, yeah, that would be an interesting way to explain the H5N1 claims. I think that's highly unlikely because it would require cooperation among scientists.
Yeah.
On something non-scientific,
Right.
and I just don't think that's going to happen. He has a postscript. With trepidation, I took the survey. This is the listener survey that we were flogging on everybody. I thought it was going to include virology questions, such as what's your favorite sequencing service, or make me feel silly for listening without a formal education in biology. Where did you get your PhD? But the questions were about if I buy stuff that I hear about on podcasts. Easy. Short answer, yes. Long answer, hell yes. Thank you very much, Tom.
Yeah, thanks for taking this survey, too. All right, the next one is from Lori Ann. Dear Twivers, they say that any press is good press, and while the hype concerning Faucher's virus is overblown and unfounded, it is heartening to know that the general public has an interest in knowing what virology researchers do and how it might impact them. It's exciting to hear students chatting about the super virulent virus and engaging in virology in ways that I can only hope to achieve in the classroom. Thought you might be interested in the attached editorial written by science writer Laurie Garrett about government regulations on synthetic biology, using Faucher's experiment as an example. Keep up the great work. I started listening to TWiV as a postdoc back when the episode numbers were in the teens and haven't missed an episode since. Laurie Ann is a professor at Marquette University, which is where our guest a couple of weeks ago was from. Michael Walsh had gone to Marquette, if I recall.
I think that's right.
Cool. So we'll put a link to that article. I'm not terribly fond of that Laurie Garrett article, but we'll put it up there for everyone to look at.
Yeah. I do think that any talk about viruses in general ends up being a good thing, because it really, the more people get interested in the stuff, and whether they're listening to TWiV or they're reading Laurie's articles or Zimmer's articles, eventually they realize there's a nugget of truth in there, and that they'll get there eventually and not be as scared as they probably are.
Yeah, I agree. I agree. All right. Looks like you're next, Alan.
Yes, it looks. What a coincidence! I'm next.
I thought you would like to.
Yes, I'll take this one. It's aimed at both of us, but
I had
Joe,
nothing to do with this one.
Right, you can recuse yourself here. Joe writes, I started listening to TWiV around number 40. I enjoyed it so much that I listened to all previous episodes and haven't missed a new one since. You managed to intrigue me so much about virology that I also listened to your entire course at Columbia twice, read several virology textbooks, and engaged in similar self-study in immunology as background to appreciate the field better. Obviously, I'm not a microbiologist. In fact, I'm just a lawyer. Gasp. Although my undergraduate degree was in organic chemistry. But thanks in large part to you, I have developed what I believe is well-informed side interest in the viral world. All of this is to say that I feel I owe you a debt of gratitude. And it is for that reason, and also so that you might take my feedback more seriously, that I'm writing you a private email as opposed to simply posting my concerns as a comment on the TWiV site. I think this is a perfectly appropriate thing to read on the episode.
Yeah,
Sure.
I mean, we're not. I mean, I think he has some good points. That's why I wanted to read them.
Yeah.
Okay.
For the first time in hundreds of hours of listening, and we're not giving last names or locations or anything. So, for the first time in hundreds of hours of listening to TWiV during TWiV 169, I actually felt like shutting down my browser right in the middle. In particular, I was completely turned off by the group's discussion of the NYAS, New York Academy of Sciences, H5N1 dual use forum. It is not that I even disagree with you on the merits of whether or not the details of the Fouchier research should be released, but the disdain and contempt the TWiVers showed for those who disagreed with them, especially Michael Osterholm, was unbecoming. I've followed the recent dual-use research debate and watched the 2-hour New York Academy of Sciences forum. Michael Osterholm might be wrong, but he struck me as sincere and entirely motivated by genuine desire to discharge his mandate to the best of his ability. Yet the TWiVers were uncharacteristically hostile to him in particular, and the other side more generally, to the point of even impugning their good faith. There seemed to be no recognition that the NSAB is charged with a horrible responsibility, one which you have the luxury not to bear, but just peck at. Instead, there was just scoffing at their consideration of unquantifiable risks as fear-mongering, and I don't think you in particular, Vince, were fair in your selective quotations and summary of the event. Your point about the case fatality rate was obviously right, but the show beat that straw horse to death. Well, if it was right, then it wasn't a straw horse. Anyway, it didn't offer much more than a caricature of the other side's arguments. You were all over Osterholm, but strangely silent about Arturo Casadeval, who was in substantial agreement with him, but also carries the sort of credentials you seem to respect more. What the TWiVers displayed in this show was an irony one sometimes sees in highly specialized science experts. They're so meticulous and careful in describing their own fields, the precise extent of their knowledge, and the limited conclusions that can be drawn from their research. They're hyper-vigilant of outsiders misunderstanding the implications of their work or making exaggerated claims about it. Yet if their interest is piqued in an area outside their chosen fields, especially in policy or political issues that might affect them or their work, they will generalize and make sweeping conclusions and pronouncements with the best of them, assuming knowledge and judgment about outside matters, like national security, that would cause them to breathe fire if an outsider did the same in their field. I've also been reading a lot of cognitive psychology lately, which has inspired me to call this phenomenon the hubris heuristic. In fact, that whole part of TWiV 169 had a polemical flavor I'm quite used to as a lawyer, but which I dabble in science to escape. How disappointing. One of my favorite things you did on your show back in the early days was to ban the occasional political witticism, usually from Dickson and usually aimed at Bush. Maybe you can renew that wise impulse at this time. Regards, Joe.
I think Joe has some good points. I agree that we're a little hostile to Osterholm, and I could justify it by saying that he insulted my PhD advisor. But you're probably right, Joe, that we were my quotations were selective and not fair, and we probably went a little over the top. So you're right, and I think that it's probably a good idea just to stick with the science. We were trying to do that at the New York Academy meeting, but it degenerated into something more than that. So I take your criticisms to the heart. I think you're right in many ways, and we'll try and temper it in the future.
And I had a blog post up. Mike Osterholm called me, and I summarized the conversation that I had with him on the phone, which was considerably more cordial than the discussion that he had at the New York Academy of Sciences with other researchers. Yeah. And I, was hoping in that blog post to help in the the toning down that we all seem to want to do, both sides of the discussion. But I'm not sure how far that really got. Yeah, it got heated, and watching the video from I was not, unfortunately, able to attend the Academy event, but watching the video from it, that was not the kind of discussion that should have been going on. It should have been more civilized than that on both sides. And yes, here on TWiV, I have participated in this as well. But I will say, I think your labeling of this as the hubris heuristic is probably a bit off base. We're not commenting on things that we have no understanding of or that are way outside our field. I spent the better part of a decade of my spare time immersed in the world of academic policy analysis when I was coaching college debate teams. And I, then got a PhD in microbiology and have spent the past almost 15 years with science policy as a regular part of my beat. This is not a general political issue. This is a science policy issue. So I would argue that I'm at least as qualified as, say, Laurie Garrett to comment on this issue and have an opinion on it. And I think Vincent, having been a flu virologist and having worked on a virus that's in the process of being eradicated, and he and I co-authored a paper that brought up many of the issues surrounding that, we're not out of our field here. I think that's taking things a bit too far. But yes, the entire discussion about H5N1 did get more heated than was productive.
I think so. I think that when you want to talk about national security, you have to understand the science, and I think a lot of the players are not. And for example, the value of animal models is often not appreciated. So right. That's where I try and bring it. And I'm sorry that it got heated. I agree, it's not the best way to go, but we'll try and avoid that in the future. Yeah. One other thing, though, Arturo Casadeval, I was all over him after the meeting. I was arguing with him and trying to understand, his thinking. So I'm disappointed in his decision. I understand it was made in good faith, but he couldn't tell me why he had come to this decision, because he said that the discussions were confidential. And I said, you have to understand, if you were in my place, you would want to know why you had come to this decision on the NSABB. So right. I'm equally critical of him, but he was more, I don't know, in some ways more reasonable on that panel than Michael was, and so that's why I picked on Michael.
Arturo did say that he started from a position of wanting it to be open and published, and he's changed his mind when he went to the meeting, which I really want. I'm interested in, I don't know if it'll ever come out, maybe it's not allowed, but what actually, who was there, what science policy people were there, what defense people were there, and what was really talked about, because it clearly, it supposedly came out as a unanimous decision, right? So it changed people's minds about when people were there. So,
I don't know. You're right. He said he changed, and he didn't tell us why. I'd like to know.
Right.
I think they found something out, maybe, and they can't tell us. I just don't know. But maybe we'll find out. I have a lot of respect for the scientists on the NSABB, and as I said before, I was surprised at their decision.
Well, and speaking of cognitive psychology, my theory is that the NSAB process was compromised by a phenomenon called groupthink, which I think they were highly susceptible to. And the fact that they reached a consensus among a group that was largely made up of scientists is, to me, kind of almost diagnostic of that.
Yeah, that's why I'm wondering what else was there. I mean, there had to be something else. So,
yeah. I mean, I just don't buy that nobody
that scared them enough to really put this put the redaction idea out there.
I just don't buy that nobody on the committee had serious reservations about that decision. And to have that come out as a consensus, that to me suggests that some strong personalities ended up leading the discussion and probably unconsciously suppressing dissent because people didn't want to get in their way.
Yeah,
right. And
that's a theory.
A strong personality, but I mean, he even said, he didn't want to be the one responsible for something releasing and then, causing havoc around the world. And maybe if they were discussing it from that position, then potentially you have to have the same conclusion. I mean, I don't think it's the right conclusion, but I think the science really does have to play a big role, and it didn't clearly didn't play a big role in that discussion.
But I do appreciate, Joe, that you're learning virology and you stuck with it, and I hope you stick with it.
We will not generally be engaging in those sorts of discussions.
We'll stay away. Can you finish up the PS?
Oh, then P.S. This is still from Joe. I was also really looking forward to the focus on viral epidemiology, hoping that the show would shed some light on some areas that I still scratch my head about, like how herd immunity is estimated for different pathogenic viruses. Maybe next time.
Yeah, we'll have Michael back, and we can
Yeah.
get back into it. That was just going to be a primer, that episode. And
and the fact that it ended up being a 2.5-hour episode tells us that we really need to have him on again.
Yes, it was really good. All right, the next and last email is from Stefan, who actually was my host out at the University of Michigan when I visited last year. He's a postdoc working on noroviruses, and he writes, I also watched the New York Academy of Sciences host a panel of leading scientists, publishers, and ethicists who discussed issues surrounding H5N1 research, which you were a part of. Dr. Fouché's research pinpointed a threat that mankind was not aware of, and thanks to their findings, we have been given the chance to act on it ahead of time. I think that Dr. Fouché's findings should be made accessible to the public and the recombinant virus be shared with select research groups. With the extensive global effort to study influenza, there may already be other labs that also introduced similar mutations into influenza without checking them in ferrets. These people need to know. The discussion whether ferrets are a good model is futile. They are only a model. They may lie. They may exaggerate. They may even be precise. We cannot possibly know, only guess. The only thing we know is if ferrets are a good model, this is very bad news. The question now is not if, but when a human transmissible H5N1 appears in the wild. May it be by nature, may it be by hand of a terrorist with a foreign or not so foreign name, or may it be by escaping from a research lab. Dr. Osterholm mentioned we cannot be wrong on the risk we take studying this virus, but what about the risk not studying the virus? Nature is the fiercest bioterrorist imaginable. Which risk is bigger, the chance of this virus escaping man-made or the virus appearing nature-made? I cannot predict the risk of the virus being released man-made, but the epidemiologists at the CDC surely can predict the risk of the virus appearing being nature-made using mathematical modeling, provided they have all the necessary information. Of course, there is a risk of studying the virus, but I sleep better knowing researchers do something about it rather than procrastinating the problem until it hits us. No one can predict the implications of research, only history will tell. The current situation is a precedent, so how do we need to learn from it? The role of government is to protect people, i.e. promoting research to develop vaccines and to provide and enforce guidelines to conduct research in a safe way. The role of the scientist is to conduct research in a responsible way, which relies on the freedom and duty to discuss findings publicly and being peer-reviewed. The role of publishers is to provide this platform. This system works if governments act on their responsibility to promote and fund vaccination research. This system works when scientists are well-educated, are peer-reviewed, and discuss their findings publicly. The system works if publishers provide editorial guidance to provide a good quality publication suited for the public. However, the system does not work if politicians publicly scrutinize vaccination and governments inadequately fund research. The system also does not work if scientists proclaim sensational research before being reviewed. And this system does not work if the press publicly discusses whether something should be published or not. Sorry, I had to get this off my chest. P.S. I Googled for a copy of the uncensored Fouché paper before the press frenzy, and there were links that claimed to contain a draft of the original manuscript. I have not verified or read any of them, but it is only a matter of time before this information gets in the wrong hands. The good thing now is that it will be almost impossible to find this among all the comments and tweets about this subject. Mission accomplished.
I looked. I couldn't find it.
You did, huh? Well, it's out there. I met someone last week who had reviewed one of the papers, in fact. So.
Oh, really? He or she allowed to talk to you about it?
Well, he wanted to know if I wanted to know the mutations. I said, no, I don't want to know what they are. But, you know, we could guess 4 out of 5 of them easily. So it's just one that he says is a surprise and that I didn't want to know about. So
And these papers, they were presented to hundreds of people, and then they've been stored on dozens of servers that are lightly secured in various academic institutions around the world and kicking around publishers' offices. None of this has been handled in a classified environment.
That's right. Yeah.
No, exactly.
So this whole discussion is about closing the barn door after the horse is already out.
Yeah, that's true. All right, that's a good letter. Thank you, Stefan. You make good points.
Has this topic brought you the longest letters that you've had since TWiV?
I don't know. XMRV had some long ones too.
Oh, that's true. Okay, maybe the two. That's true.
All right, let's do a couple of picks of the week. I think we have more in the pipeline. This will go on for a while, but that's fine. We want to be a forum for discussion too. Matt, what have you got for us?
So my pick was there's an ASM biodefense meeting, and there's going to be a special panel on the topic we just talked about, the H5N1 and the NSABB. So Fouché is going to be there, and 3 or 4 of the big players. So it's going to be live. Supposed to be live on the web during the conference. I think it's at 7:15 a.m. or something, but I think it's going to be a kind of interesting panel to talk about. So it's Osterholm, Fauci, Bruce Alberts, the editor of Science, and Ron Fouché at 7:15 a.m. on the 29th of February. So cool. Everybody can listen in and see what actually happens live at the meeting. So I think it'll be interesting to listen.
Cool.
It should be noted that today Bruce Alberts said that unless a way is arranged for disseminating the H5N1 findings, he will publish them in full because he thinks it's important that researchers get them. So it'll be interesting to hear his comments. Alan, what do you have?
I have the Imagine Science Film Festival. So Imagine Science is a nonprofit organization that I gather a couple of filmmakers with scientific interests and scientists with interest in filmmaking got together and started this thing up. They're based in Brooklyn, and they are all about making films about science. And so they, in the spirit of promoting that, I think back in about 2008, they started having a film festival that's just for films about science. And most of these are collaborations between researchers and filmmakers. And they are now, well, they're taking submissions for the next film festival, but it has to be in by March 15th. So if you aren't already shooting, you probably have missed the chance this year. But if you are a filmmaker and you want to run right out and shoot something interesting that's science related, then this would be a good place to submit it. And they've got their information on previous winners of this and a whole bunch of other interesting information on their site.
Nice. That sounds good.
Yeah.
Looking at the people here who are involved, I don't see any scientists.
They've got a couple of postdocs. I gather a lot of their staff is either part-time or volunteer.
Yeah.
And so it's film people who've done a little science training, and then there's a postdoc or two in there.
Here's someone you might know. This young lady, the communications officer, got her degree in science, health, and environmental reporting from NYU.
Yes, she's a SHRP graduate. I think she was there after I had moved on.
Emily Driscoll. Cool.
And I like this is one of those things where it's about communicating science, right? So the more people that can get a piece of this and out of sight of, without having a PhD is all the better.
Yeah. Here is a postdoc at Rockefeller who was working on how the innate immune system fights viruses. She is the producer and program director. Cool.
Very good.
Talk them into coming and filming TWiV.
Yeah.
All right. Yeah, why not?
Sure.
I have a pick from Dickson. He left me a book. It's called The Golden Wand of Medicine, A History of the Caduceus Symbol in Medicine.
Huh.
You guys know what a caduceus is?
Yes.
Yeah, so it's that...
That's, what worm infection is that based on? Is it...
it's guinea worm.
Guinea worm. That's it.
Dracunculiasis. Yeah.
Dracunculiasis.
It's a fiery serpent or something like that because it burns your leg. Dickson has been writing a book on parasitism and The last chapter was on the guinea worm, and so he went and bought this book to provide background.
Huh.
And this staff is a staff with wings, and it's got two serpents going around. It's kind of like Hermes' staff. And this is a book with a lot of background, probably more than you would want. But it's his pick. And we actually talked about this on TWiP yesterday, which will be out sometime next week. So for the lighter version, you could check out that TWiP, The Golden Wand of Medicine by Walter Friedlander. And my pick is a publication of the American Academy of Microbiology. They put out these free publications that are typically the results of committee reports. And this one is pretty cool. It is called E. coli, the Good, the Bad, and the Deadly. And it came out last year. It's all about E. coli, which, as you know, was in the news. Well, it's often in the news when strains are isolated from food that make people sick. Like last year, the O104H4 strain in Germany from sprout seeds. So this is a summary of E. coli and all that it can and can't do. So it's pretty neat.
Cool.
Check it out.
Very
It's
good.
basically written as a FAQ, frequently asked questions. What is E. coli anyway? How has it contributed to our understanding of biology? What does it do in our GI tract? So, pretty cool.
Very good.
Neat.
You're sick, and there we go. We have a listener pick of the week from Daniel. Great podcasts. I've listened to them all. TWiV, TWiP, TWIM. Insert required adulation. I enjoyed this and figured you might as well. It takes a bit to load, but it's worth it. It's a very cool thing. You got to just wait till it loads. The scale of the universe, it takes a while.
Takes a couple of minutes.
So be patient, folks. It's worth it.
Yeah.
Did you look at this, Matt?
I did. No, it's very cool.
It's cool.
Again, science for the public is good.
Yeah.
And thank you all for donating so much of your time for all of our entertainment and education. We also have a pick from Stefan, whose letter we read earlier. He said, I'm still doing some research for a polio lecture and stumbled across this wonderful video about Tom Francis and Jonas Salk. I think this clip reminds us of the forgotten perils of pre-vaccination times and puts things in perspective. If you find a public relations contact for Delta Airlines, they may want to replace some of their more recent videos on vaccination. Hail to the polio pioneers. Thank you, Stefan. That is pretty cool. It's a good movie. So we have wonderful gems found by our listeners. It's just great. That will do it for TWiV 171. You can find TWiV on iTunes. And if you like it, you can help us out by leaving a review there if you haven't done so already. It helps keep us on the front page of the Apple Podcast directory, and they get reviews that bumps us up there, and that's how new people can find us. We also are on the Zune Marketplace and microbeworld.org, where you can find an app to stream the episodes to your smartphone or Android device. We also have a Facebook page, facebook.com/thisweekinvirology. We love getting your questions and comments. Please send them to TWiV at twiv.tv. Dickson Despommier, of course, who is no longer in studio, is at verticalfarm.com, medicalecology.org, and trichinella.org. Thank you, Dickson. Insert required response. Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
And you're back on the Twitter, Alan Dove.
Yes, I am.
Are you enjoying the Twitter?
Yeah, it's all right.
It's all right.
I had been on it early on when it was first becoming popular, and then I got kind of sick of it and quit. And then now I'm back.
Good to have you. Matt Frieman is down in Baltimore at the University of Maryland. Good to have you again.
You too. It's very nice to talk to you guys.
It's your third visit on TWiV.
I know. I need like a little hash marks on the back of my desk for all the episodes.
Well, it's fun to have you. We like having you. Anytime, you're always welcome.
Sure. And I do have to say, I think you guys you do a great job. And I hear it from everybody when you visited here and all the students that hadn't listened to TWiV until you came. So I think you guys, it's pretty remarkable you have this many episodes through the years.
It's a lot of episodes.
I think it's not an easy thing to do every week. So I do appreciate it as a virologist and
thank you.
just professor in general.
Thank you.
It's a lot of fun to do it. I don't even view it as work, right? Right, Alan? Do you view it as work?
No. This is a kind of a break. I mean, it's got some work aspects to it, but
You learn a lot.
a lot of fun, and it's incredibly educational.
Yeah, I have learned a ton. I would, you broaden your views when you do this because normally you're pretty focused in virology or whatever field of science you're in, right?
Right.
Yeah, and I read a lot of different fields of science, but I'm never reading them for very long.
It's
I'm
true.
I'm not usually doing really deep dives in them or spending an hour interviewing one researcher.
Yeah. So Matt, by the way, I had a conversation with Carl Zimmer the other day, and he mentioned that he had visited you. Was that
He
fun?
did. It was
that
great.
fun?
It was
Yeah.
really nice to meet him in person, actually.
So you had a seminar basically visit for him. Is that how it was set up? And he talked with faculty and so forth, or what?
Right, exactly. The same way a normal, a standard scientific, scientist would come down.
Yeah.
We brought him down. He talked to, a handful, 8 or 10 faculty during the day and gave a seminar on viruses and science writing and phage and history of HIV and, kind of all of, he had written a book called History of, what's it called? A Planet of Viruses. So we brought him
Yes.
down when that had come out. And so it was great. It was really nice to meet someone who does this kind of the outward public science for a living.
Yeah.
So it was really nice to get him down here, and he will go to other universities too. So if anyone's out there that wants him, I would say tweet him or email him and try to set something up. It was great.
Yeah, it's a good idea. I think we should do that here because there are a lot of people who probably are thinking about careers in communication, and it would be good to hear him.
Absolutely. And one plug for our micro program down here, the University of Maryland School of Medicine has a very good microimmunology graduate program. We're doing our last day. Today's our last interview day for the year for the new grad students. But if any undergrads out there or anyone else is interested, please look at the University of Maryland website, find their grad program, or email me, and I'll get you in touch with the right people. It's a good place to be, I promise.
Cool.
It's good. I liked it. When I visited, I thought it was good. Good
Good.
department, good city. You going to be in ASV, Matt?
I won't actually be this year. We are expecting a son, another child in May.
Wonderful.
Congratulations.
So my wife has put the stop on the summer conferences. So I'll miss ASV this year, but I'll be listening on TWiV for the next week.
Great. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.